My name is Ivania Valdés. I'm a PhD student of the Integrative Genomic Program at Universidad Mayor, but I'm currently working in Pontificia Universidad Católica de Chile. I'm working in Chile actually, and where I'm developing and finishing my PhD thesis, which is correlated with the research on the tumor microbiome implications in the lung cancer, specifically with adenocarcinoma. And we wanted to know which part of the bacteria present within the tumors are interacting with the host in describing the mechanisms that are involved in this relationship. So with that information, using all of this bioinformatic approach that I'm working on, trying to understand what is happening, and maybe with that information took some of those mechanisms and maybe prove it in like an experimental studies. The abstract that we sent it to the ACR, actually I sent it as a poster and it was selected to a mini-symposium, which was very great. I'm very grateful. And I explained it like various little things that I already done in my thesis, that is the use and the integration of different omics, and at this point I'm using 16S RNA sequencing, proteomics and metabolomics, and to trying to answer and to describe this microbiome composition in the tumor of adenocarcinoma and these components such as peptides and metabolites associated with bacteria. And we actually found very interesting results. We found an increasement of some potentially pathogenic bacteria within the adenocarcinoma tumors, and some metabolites are already been described as an immunosuppressive metabolites and promote this state in the patient. So it's very exciting. There's already been described some bacteria within the lung microbiome and the tumor microbiome in lung cancer. However, the exact parts of these bacteria have not been clearly defined. So what we found was that some metabolites they may be implicated in this promotion of different states, such as immunosuppressive or immunoevasive response at the immune system response. So we think that they are very exciting results, since you can take this to prove it in vitro or in vivo to check that they are promoting these states and actually contributing to a worse disease outcome for the patients. Well, what we want to try to do is that actually finishing to the another omic technique that we need to do is transcriptomic, to see the gene expression of some of the proteins that we already found and the metabolites. And we wanted to take like some of those potentially pathogenic bacteria that we found, such as Pseudomonas and other metabolites, to test it in vitro with some human cells, lung cancer cells, and then maybe translated that into an in vivo analysis.